Good morning. My name is Parisa Yousefpour, and I'm a postdoc fellow in Dr. Darrell Irvine's research group at MIT's Koch Institute. And in this presentation, I will share with you guys our recent and also current work on using self-replicating RNA or replicon technologies for cancer immunotherapy and also for vaccine development. So currently, there are three main nucleic acid-based modalities for gene therapy: DNA, conventional mRNA, and self-replicating RNAs or replicons. DNA is required delivery to the localization to the nucleus for homologous recombination, so this adds a delivery barrier for their application. RNAs, either the traditional mRNA or self-replicating RNAs, they only need to be delivered to the cytosol for protein expression. With mRNA, we could deliver high quantities of it to the cells, but the problem with traditional mRNA is that gene expression from traditional mRNA is usually very short-lived, and it goes away within a few days maximum. Replicon, on the other hand, it self-amplifies inside the host cells. So it can provide a high level of gene expression from a small initial dose, and it also can sustain the gene expression over an extended period of time for up to a month. And finally, replicons have intrinsic adjuvant activities. The double-strand RNA that is formed during the replication of replicon, it activates the pattern recognition receptors and stimulates the innate immune signaling pathways. The replicons that we work with in the Irvine lab in collaboration with the Weiss lab at MIT are derived from the alpha virus genome. The original virus genome is shown here. It encodes for non-structural proteins and structural proteins. Non-structural proteins include proteins like helicase and polymerase that form the replicase complex that is necessary for the replication of the replicon. Structural proteins include like capsid proteins and glycoproteins that form the virus trimer. So in making the replicon, we remove the structural proteins and we replace them with our gene of interest. So the non-structural proteins are still there, so our replicon is still able to self-amplify. The biology of the replicon is shown here. After transfection into the host cells in the cytosol of the cells, the non-structural proteins are translated. They form the replicase complex. This replicase complex then makes a negative strand from this original positive strand. And again, the replicase complex makes positive strand copies from this negative strand, and it also catalyzes the transcription of the subgenome. And translation of this subgenomic transcript then results in the expression of our gene of interest, which could be like antigens, cytokines, or other immunomodulatory payloads. So we use the firefly luciferase or FLOC as a reporter protein. And then we made replicons that encode for FLOC. When these replicons were injected in mice, and bulb C mice by intramuscular injection, we monitored the FLOC expression by bioluminescence imaging. And we observed that with replicon platform, it provided like gene expression that lasted for about a month, and the expression was longer and also higher compared with mod RNA, where it allowed for gene expression for only 2 days. So a key issue for RNA therapeutics is their transfection into the host cells, specifically the cytosols of the cells. Lipid nanoparticles, or LNPs, are non-viral genome vectors that are commonly used for addressing this issue. Lipid nanoparticles usually contain a cationic lipid or an ionizable lipid. This ionizable lipid is positively charged at low pH, but is neutral at physiological pH. So this pH responsiveness enables this lipid to condense and encapsulate the replicon at low acidic pH and protect the RNA and deliver it into the cells at physiological pH. And finally, it allows it to it facilitates the endosomal escape of the RNA, and it's released into the cytosol at the low endosomal pH. We are working with the Dong lab at Ohio State University to explore and also optimize the lipid nanoparticle formulations for different applications, also for transfecting different cell types. To make our replicon LNP formulation, we use a microfluidic device. Here, we have an aqueous solution that contains our replicons and an organic solution that contains our lipids. These two solutions are injected into the microfluidic device, and they're mixed rapidly together and give us our replicon LNP product. So with this method, we have been able to make LNPs with a diameter about 50 nanometer, as was confirmed by TEM and DLS methods, and it also provided over about 90% RNA entrapment efficiency. So here I'm gonna present two recent publications from our lab for developing replicon LNP therapeutics, one on the cancer immunotherapy side, the other on the vaccine side. So for the vaccine immunotherapy, we are interested in using replicon LNP formulations to develop in situ vaccination strategies. In these strategies, the aim is to remodel the tumor microenvironment and initiate immunogenic cell death in the tumor. And this results in the release of tumor antigens that are then trafficked to the lymph node by antigen-presenting cells. And in the lymph node, these antigens prime de novo T cells, and these prime T cells are recruited then to the primary tumors or also to the distal metastatic sites. So this provides a systemic this local therapy can provide a systemic immunity against cancer. And I should also mention that one advantage of this in situ vaccination strategy, the main advantage, is that, as you can see, it uses the tumor itself as a source of antigen. So it obviates the need for identifying a specific tumor antigens for each tumor type. So in our work, we constructed replicons that encode for the inflammatory cytokine IL-12, fused to albumin and also lumican. Lumican is a collagen-binding protein. We added it there to help keep the IL-12 fusion bound to the ECM and retain it within the tumor to limit its toxicity. These replicons were encapsulated in LNPs, and we aim to target multiple synergistic pathways of the immune system in this strategy. We intentionally used a cytotoxic LNP to initiate the immunogenic cell death of the tumor cells. The replicon itself, it triggers type 1 interferon responses, and it stimulates innate immune signaling pathways. And finally, the IL-12 payload, it can remodel the tumor microenvironment, and it can recruit different immune cell types like T cells or NK cells and activate them in the tumor. So therapeutically, when these replicon LNPs were injected intratumorally in B16 melanoma tumor model, they resulted in elevated levels of IL-2 in tumor, which lasted for at least 7 days. They also induced exposure of calreticulin on the cancer cells, and calreticulin is a hallmark of immunogenic cell death. Type 1 interferon was also increased in these tumors, and finally, when we look at the immune composition of the leukocyte in these tumors, our formulation was able to result in great infiltration of CD8 T cells and granulocytes into the tumor. So in terms of toxicity, our replicon LNP formulations showed a transient toxicity as indicated by the weight loss in animals. And when we look at the tumor growth, a single injection of these replicon LNP formulation was able to eradicate the primary tumor, and also interestingly, it also slowed down the growth of the untreated tumor. So it was able to create the so-called abscopal effect. And the survival of the animals was also prolonged with these formulation. So this work shows us that local therapy, local injection of our IL-12 replicon LNPs can generate a systemic immunity, and this is critical for which is very critical for the translation of this technology for treatment of metastasis disease in humans. So now moving to vaccine side, here we constructed replicons that encode for an HIV immunogen. When these replicons were injected in bulb C mice, they were able to elicit antibody titer even following a prime injection. And this antibody titer was further enhanced by boosting. In addition, our replicon platform was able to elicit different subclasses of antibodies, IgG1, IgG2A, and to a lower extent, IgG2B. And when we look at the germinal center responses, which are more indicative of the quality of the immune response, we see that with replicon immunization, it resulted in higher percentage of immunogen-specific germinal center B cells compared with protein immunization. In addition to the mouse model, we also tested these replicons in rhesus macaques as non-human primates. And consistent with our murine data, we again observe antibody titers and germinal center B cell responses with these replicons, which highlights the translational potential of this platform. So now, building on these recent works in our lab, what we are currently working on in collaboration with the Weiss lab at MIT is to incorporate synthetic biology tools to develop next generation replicons where we control the location and also the timing of gene expression from these replicons. For regulating the location, we use classifier gene circuits that enable cell type specific gene expression. And for regulating the timing, we use a small molecule responsive gene circuits where we can control the timing and also the level of gene expression from these replicons with a small molecule drugs. So first, about the classifier circuits. So classifier circuits are based on incorporating microRNA sensor that were actually discussed in the previous talk in our replicon platform. So for example, in KP lung cancer cells, the microRNA or miR-194 is highly expressed, but miR-150 is present at low levels. So if we design replicons using mVenus as the fluorescent reporter protein with target sites with either of these microRNAs, with classifier with miR-194 classifiers, we observed that gene expression was reduced, while with miR-150, the gene expression was not impacted as much. So this kind of suggests that with miR-194 classifier, the classifier is being degraded in the KP lung cancer cells. So in another proof of concept experiment, we designed dual cargo replicons that encode for mVenus and mKate reporter proteins. Here we added a target site for the synthetic siRNA FF4 after the subgenomic promoter for mVenus. And when we tested this replicon in BHK cells, as expected for MK, its expression was independent of the FF4 siRNA presence. But with M. venus, this expression decreased by over 5-fold in the presence of this FF4 siRNA. So you can imagine that this platform can be very powerful for cancer immunotherapy. With this platform, we can have a single replicon that encodes for different payloads, like different cytokines or cytotoxic proteins and other stuff, and with different target sites. And when they are injected in the tumors, the same replicon is delivered to different cell types, T cells, cancer cells, or dendritic cells, and as such. And in each cell type, depending on a specific microRNA, a specific payload will be expressed. So that was about regulating the location of gene expression. Now, the other, as I mentioned, another avenue we are interested in is regulating the timing of gene expression with small molecule drugs. One example application that I'm going to share with you is to use this platform to control vaccine kinetics and study how the expression patterns of antigen impacts the immune response. So this is motivated by a previous work by our lab. In that work, with using protein immunogens, it was shown that the kinetic of antigen pattern matters in vaccination. So when the same dose of antigen, the same total dose of antigen was injected in exponentially increasing doses, it resulted in higher antibody titers compared to when it was injected in constant doses or in constant dose or de-escalating doses. So, but you can imagine that this repeated dosing regimens is not practical for human vaccines. So now the question is that how we can implement these dosing regimens in a practical vaccine. So since replicon provides gene expression over an extended period of time, we believe that it can provide an ideal platform to implement different antigen dosing patterns. So for this, we use a small molecule responsive gene circuits. The gene circuits that we have started with is responsive to trimethoprim or TMP, which is an FDA-approved small molecule drugs. This circuit is based on the destabilization domain from bacterial dehydrofolate reductase. This destabilization or domain or DD is degraded quickly by proteasomes unless it's stabilized by TMP. So now if we fuse our protein of interest, like in the case of vaccination antigens, to this DD, the instability of DD is confirmed to the antigen, and the whole fusion is degraded by proteasomes. But when TMP is present, it stabilizes DD and it turns on our gene expression. So to incorporate these TMP responsive gene circuits in our replicon platform, we took two approaches, payload fusion and NSP fusion. In payload fusion, we fuse DD directly to our gene of interest. As I mentioned, without TMP, the whole fusion will be degraded and gene expression is turned off. But with TMP, the fusion is stabilized and gene expression is turned on. In NSP fusion approach, we target the replication mechanisms of the replicons. Here, we fuse the DD to the non-structural proteins of the replicons. When there is no TMP, the replicase complex is degraded. The replicon is not able to copy itself. It decreases the copy number of the replicons and turns off the gene expression. But when with TMP present, this replication machinery is restored and our gene expression is turned on. So first, we fused our DD to different NSPs of the replicon backbone to identify the best NSP to regulate. In vitro, we found out that the NSP3D diffusion provide the best regulation. It provided over 100-fold difference between on and off state, and also the off state was close to the background, which indicates non-leaky regulation. We also tested these circuits in vivo in mice. So here we tested 3 circuits with DD fused to NSP3, fused to FLOC or to both FLOC and NSP3. And when these circuits were injected in mice intramuscularly, and TMP was provided in the diet. So checking the bioluminescence, all these 3 circuits showed regulation with higher gene expression in the presence of TMP compared to that in the absence of TMP. And our double DD circuit turned out to be the best performing one, as at day 5, it provided about 50-fold difference between on and off state and an off state close to background. So we then use this NSP3 DDE circuit for regulation of the expression of an HIV transmembrane trimer. So in vitro, we checked the expression of this transmembrane trimer on the cell surface by flow cytometry and confirmed that the expression could be regulated by TMP. And for our in vivo experiment, we tested different TMP dosing regimens. I'm showing 3 of the regimens here. First group of animals received no TMP in their diet. The second group of mice were put on diet supplemented with a low dose of TMP, and the third group was put on low TMP dose for 2 weeks, and then it was moved to a high TMP dose to kind of like create an antigen boosting effect. So as you can see from the antibody titer, our no TMP group showed the lowest antibody titer among all the groups. And interestingly, our kind of TMP boosting strategy seems to have worked, and it provided higher antibody titer compared to the constant low dose of TMP. So we were very excited by this data, as we are showing here that the antigen expression and also the output immunity in vaccination can be regulated by a small molecule drug. So with this, in summary, I hope that I have convinced you that self-replicating RNAs are promising technologies for cancer immunotherapy and also for vaccine development, and that we can integrate synthetic biology tools in this replicon platform to control the location and the timing of gene expression, and to program kind of more sophisticated therapeutic interventions. And as it goes with research, it's a collaborative effort contributed into by many former and current lab members. We're also very lucky at the Irvine lab to be collaborating with many awesome groups in and outside MIT. And thank you guys for listening.
Thank you for this interesting presentation. So you got a couple of questions here. Shall I read it to you? Or?
Yeah, I can read. Yeah.
Okay, let's start with one with 2 votes. How do you control dosing of the RNA? Will initial dosing errors be amplified or normalized due to the replicon?
So when we talk about dosing, we are just talking about the dose of RNA that we are injecting to the mice. So that's kind of, and then, I mean, that's why we have control groups, and to just, I guess, factor in the, I guess, inconsistencies in dosing, and then we, I guess, doing our statistical analysis to rule those out. I'm not sure if that answered the question.
And then expression level, stability, copy numbers versus circular RNA.
So our replicons cannot be made in the circular form. Traditional mRNA can be made, and there are reports showing that the circular form can provide higher expression levels, but with replicons, because of its self-application mechanisms, it's not possible so far. So.
What are the critical aspects, differences of your lipids, LNPs, lipid nanoparticles compared to today's messenger RNA vaccines?
So what we like to claim is that our replicons provide gene expression for a longer time. So that's the biggest advantage that it can provide compared to today's mRNA vaccines. Today's mRNA vaccines, like I said in my presentation, the gene expression usually rapidly, the antigen expression rapidly goes away within a few days. If you consider natural infections, in natural infections, the infection lasts for over a long period of time, and we believe that's why that natural infection leads to such a robust immune response and germinal center responses. And so we believe that with replicons, we can better mimic that natural infection by providing longer expression of the antigen.
How long do you think replicons would be effective for in humans?
So how long, in terms of how long the antibody titers last, so our best models so far are the non-human primate models, and those, we see antibody titers for a couple of months after immunization.
Oh, wow. Good, and then the final question, can you use your circuits that express only particular cells as targeted therapy for cancer? Because local injection tumor may not be always possible.
Can you? So that's what we are trying to achieve with our classifier circuits to enable cell type specific expression of our replicons. But as I said, our main efforts are on local injection of replicons first to limit the toxicities, and second we believe that we can achieve systemic immunity by local injection.
Good, and I think there's another final question. Self-replicating RNA technologies for vaccines, cancer, and immunotherapy. That was your title. So, thank you very much.
Yeah.